Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is meant for 21st November 2020. As all of us know that we are into the revision mode and we are revising the entire syllabus of 12th biology. As part of it, we were revising chapter number 12, biotechnology and its applications. In our previous session, we were talking about biotechnological applications in agriculture. And in that, we were talking about transgenic plants, role of TI plasmid, and role of BT, Bacillus thuringiensis. And in this session, we are going to talk about the biotechnological applications in medicines. And you must also remember that we were talking about the how to silence the mRNA, silencing of mRNA in order to prepare pest-resistant plants. So with that in our background, we move ahead with biotechnological applications in medicine. The rDNA technological process, recombinant DNA technological process, have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Why do you require safe and more effective? As such, whatever chemical medicines we are using in the form of tablets, capsules, injections, no doubt they have side effects on us. If the medicine that we are taking is for stomachache, let us say, we will be getting relief from stomachache, but dehydration sets in. So here our purpose in medicine field through biotechnology is that whichever area has got pain or whichever area is to be corrected, only that area will be touched and no side effects of other organs or tissues. That is the purpose. So at present, about 30 recombinant therapeutics have been approved for human use world over. In India, 12 of these are presently being marketed. So one of the most important medicines that we are getting in India as well as worldwide is genetically engineered insulin for human beings. Since it is insulin for human beings, it is called humulin. So, through biotechnology, we are able to synthesize this humulin, and this is of great use for all the diabetic patients. So, how is to be done? Now, basically, we are talking about the insulin, and whenever we talk about the insulin, in human beings, insulin is secreted naturally by pancreas. So, in times of pancreas not functioning properly as expected, there will be insulin deficiency. Whenever there is insulin deficiency, blood glucose levels will not have control. As a result, sugar or glucose will be passed out of our body through urine. As the person loses all important energy giving glucose through urine, person becomes weaker and weaker. And this results not only in making the person weak, if there is any injury to that diabetic person, to heal that injury, it takes a lot of time compared to a normal healthy person. So adult onset of diabetes can be controlled by taking insulin at regular intervals. So once the pancreas is not working, then insulin deficiency takes place naturally. To control that blood sugar levels and to control the diabetes, artificially insulin has to be given to the person, to the diabetic person. Now so far, small doses of injections were given, and sometimes you must have observed some people, diabetic people, taking themselves injection. And nowadays, of course, even tablets are available, insulin tablets. So people who are averse or scared to take injection, they are taking even the tablets. So the main source of this insulin was isolation of insulin from animals. Nowadays, insulin can be obtained from bacterium using techniques of biotechnology. This is the most important one. Insulin was earlier extracted from pancreas of slaughtered cattle and pigs. But insulin from these sources develop allergy or other types of reactions to the foreign protein. Insulin consists of two short polypeptide chains, chain A and chain B. And these chain A and chain B are interlinked together by disulfide bridges like this. So this is proinsulin, and you are seeing beta cell peptides, and you have got this A peptide and B peptide. This A peptide and B peptide have got a disulfate bonds, and there is a free C peptide. So in humans, insulin is synthesized as a prohormone, which contains an extra stretch of chain of peptides called C peptides, which is a free one, free. This is absent in mature insulin. So the difference between proinsulin, as you are seeing with the colors, you got a chain, A peptide chain, plus B peptide chain, plus C peptide chain, and that is proinsulin. When it becomes active insulin, what's happening is peptide A peptide chain and B peptide chain are interlinked with disulfide bonds, and the C peptide chain gets freed, it becomes independent. Once the C peptide becomes free, then the proinsulin is said to have become active insulin. So the main challenge for production of insulin using rDNA technique was getting insulin assembled into mature form. That means how to make this C peptide free. That is a challenge for us. An American company, Eli Lilly, in 1983 prepared two DNA sequence corresponding to A chain and B chain of human insulin and introduced them in plasmids of E. coli to produce insulin chain. Chain A and chain B were produced separately. See, this is important. Separately, extracted and combined by creating disulfide bonds to form human insulin. So this human insulin is called humulin. So here you are seeing it, ATG, TGA, synthetic A chain, gene 163 nucleotides, and synthetic B chain having 90 nucleotides. So now there is a combination A and B, and that is being made a plasmid. You can see here the genes and the cut portions of this as well as this, and this is attached to the trans. Once the rDNA is prepared with the attachment of desired gene into the plasmid, transform E. coli, so fusion of proteins produced. So you got the A chain as well as B chain. Now cleave the residue with cyanogen bromide, and once that is done, now we are able to do the attachments of A chain with B chain, and you can see A chain getting interlinked with B chain with disulfide bonds, and that is active insulin. This is how we are able to make active insulin. So basically, the applications in medicine is the recombinant DNA technological process have made immense impact in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce, this is important, do not induce unwanted immunological responses as is common in case of similar product isolated from non-human sources. At present, about 30 recombinant therapeutics have been approved for human use over the worldwide. In India, 12 of these are presently being marketed. Initially, E. coli is a bacterium, good bacterium present in our small intestine, was used as the host for obvious reasons of ease in cloning. But yeast is fast becoming the host of choice for production of recombinant proteins. In 1921, Banting and Best, along with MacLeod, succeeded in preparing pure extract of insulin, thanks to these three gentlemen, from the pancreatic isolates of dog. They demonstrated that administration of insulin could cure diabetes in human beings. Later, insulin for curing diabetes used to be extracted from pancreas of slaughtered pigs and cattle. The insulin isolated from other animals is just as effective as that secreted by the human body itself, and it would not elicit an immune response in the human body. However, insulin from an animal source caused some patients to develop allergy and other types of reactions to the foreign protein. Genetically engineered insulin was the solution to this problem. As all of you know, insulin is made up of 51 amino acids. This is 51 amino acids arranged in two short polypeptide chains. So there is a chain A, and this has got 21 amino acids, chain A, and chain B having 30 amino acids. So chain A and chain B together 51 amino acids, 30 plus 21. And these are interlinked together by disulfide bridges. So sulfur bridges, disulfide bridges. In humans, insulin is synthesized as a prohormone which contains an extra stretch called the C-peptide. So these are interlinked with the disulfide bonds, okay, A and B, and then there is a free C chain, and this is a freed one. It's not connected. And this stretch is called C-peptide. The freed one is called free peptide with 33 amino acids. 33 amino acids. This C-peptide is not present in mature insulin as it is removed during its maturation. The main challenge for production of insulin using rDNA techniques was getting insulin assembled into a mature form. In 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in plasmids of E. coli to produce insulin chains. Chain A, chain B were produced separately, extracted and combined by creating disulfide bonds to form insulin. And since it is related to human insulin, abbreviated as humulin, Humulin. This is how human insulin is synthesized through biotechnology. So basically what we require a bacterium, and inside the bacterium you got a bacterial DNA with a plasmid DNA, and the plasmid DNA is cut with restriction enzymes, and once that is cut, the gene of interest is separated, and we take human pancreatic cell. In that human DNA is there, and in human DNA we identify the gene that produces A and B peptides. We cut that portion. So the cut portion of a human insulin producing gene and the gene of interest from the plasmid of bacterium E. coli will be ligated, and that's how we get annealing of DNA ligase. Once the annealing is done by DNA ligase, we get a rDNA, and this rDNA is introduced into the bacterium. Once that is done, insertion of rDNA with the gene of interest into the bacterium, now recombinant bacteria multiply and produce human insulin, either peptide A or B, depending upon the gene cloned in the fermentation tank. So, and we put this in the fermentation tank, and human insulin is extracted, purified, and packed into the bottles. So this is how human insulin is produced. And you just see how the biology in general, biotechnology in particular, is coming to the quick aid of our human beings in the field of health. And all of you know, health is wealth. So be healthy. Take